photosynthesis captures energy from light and uses it to drive the synthesis of carbohydrate from carbon dioxide in water. It uses different reactions to achieve the opposite process of cellular respiration. To capture this energy, the cell uses chlorophyll. There are different chlorophylls which vary on the wavelength of light they absorb. They are composed of a hydrocarbon tail attached to a light-absorbing active site centered around magnesium. The photosystem consists of light-harvesting matrices made from chlorophyll and other pigments in a protein matrix. The photosystems are embedded in the thylakoids. In prokaryotes, the thylakoids are invaginations of the cytoplasmic membrane. In eukaryotes, they are formed from the folds of the inner membrane of the chloroplasts. Here we have an overview animation of photosynthesis.
The process of photosynthesis produces ATP from ADP and PI by using the energy from light to excite electrons that are passed along an electron transport chain. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. The following complexes are found in the photosynthesis electron transport chain: photosystem 2, cytochrome B6F, photosystem 1, ferredoxin NADP reductase, and the complex that makes ATP, ATP synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP and the electrons to form NADPH, and ADP and PI, which combine to form ATP. Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem 2 complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem 2, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. 1 photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem 2 are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient, similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the photosystem 1 complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP reductase, also known as FNR. After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP, NADPH, and molecular oxygen are the final vital products of photosynthesis.
In photosynthesis, we have light-dependent and light-independent reactions. The light-dependent reactions involve absorbing energy from light and then using redox reactions to store the energy in ATP and NADPH, making them dependent on light. The light-independent reactions synthesize glucose from carbon dioxide and water. They are not dependent on light because they use ATP and NADPH to fuel the process, which was originally obtained from the light-dependent reactions. This animation shows the light reactions of photosynthesis. Step 1, we have the electrons flow to NADP+. Step 2, we split water. Step 3, we have the proton pump. And step 4, ATP production. The light-dependent reactions occur in all photosynthetic organisms. In cyclic photophosphorylation, the photosystem 1 pigments will absorb light energy and transfer it till it arrives at the reaction center chlorophyll. The photosystem can almost be thought of as a natural solar panel absorbing the energy from light. It does this by passing down the electrons, and the energy is used to pump hydrogen ions for the proton motive force, similar to what is seen in the electron transport chain. The final electron acceptor is the original reaction center chlorophyll, which donated the electrons. The hydrogen gradient drives phosphorylation. It is cyclic because the electrons are returned to the original reaction center and can be reused. Non-cyclic photophosphorylation occurs in all plants and algae and in some bacteria. It requires photosystem 1 and photosystem 2. First, light excites the photosystem 2 and the electrons are passed down to photosystem 1. Photosystem 1 is energized with additional light and NADP is reduced to NADPH using the same proton motive force. The NADPH is used to fuel light-independent reactions. This uses a different final electron acceptor, so it must constantly replenish electrons to the center of the photosystem 2, making it non-cyclic. Here we have an animation of the types.
In bacterial photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes to ferredoxin, and then down through the cytochrome B6F complex, plastocyanin PC, and finally back to the reaction center. The energy released during this electron transport generates a proton gradient which is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation. The reducing power needed for biosynthesis is not generated in the process of cyclic photophosphorylation. Plants and cyanobacteria utilize two photosystems which work sequentially to produce both energy and reducing power. First, a photon of light ejects a high-energy electron from photosystem 2. The electron lost from photosystem 2 does not return to photosystem 2, but is replaced by an electron generated from the splitting of water and the production of oxygen. The electron then travels from the excited reaction center of photosystem 2 to plastoquinone, to the B6F complex, to plastocyanin, and finally to the reaction center of photosystem 1. This electron transport system generates a proton motive force that is used to produce ATP. Since the excited electron does not return to photosystem 2, this mechanism for making ATP is called non-cyclic photophosphorylation. When photosystem 1 absorbs a photon of light, it ejects a high-energy electron. The energy from this light absorption is used to generate reducing power in the form of NADPH. The ejected electron is replaced by an electron from photosystem 2.
In the light-independent reactions, carbon from carbon dioxide is joined to form carbohydrates. In carbon fixation, one carbon from carbon dioxide is attached to ribulose 1,5-bisphosphate, or RUBP, with 5 carbons to make the 3-carbon 3-phosphoglycerate. 3 molecules of RUBP combine with 3 carbon dioxides to yield 6 of the 3-phosphoglycerate. The RUBP is derived from the precursor metabolites produced in the pentose phosphate pathway, so the substrate is regenerated. In the Calvin cycle, or Calvin-Benson cycle, for every 3 carbon dioxides that go into the cycle, 1 molecule of glyceraldehyde 3-phosphate leaves. For every 2 glyceraldehyde 3-phosphate that leaves, they go through the reverse of glycolysis to make glucose. So this is an animation of the Calvin cycle. There are two main pathways that are very common in the microbial world. However, there are numerous other pathways allowing bacteria to produce and consume numerous other substances, ranging from methane to sulfur compounds and many other things. We could fill an entire class looking at metabolic pathways alone. Amphibolic reactions are those which proceed in either direction towards catabolism or anabolism. In carbohydrate biosynthesis, glucose 3-phosphate is used as a starting point for synthesis of several sugars and polysaccharides. Gluconeogenesis, which literally means making new glucose, is the synthesis of sugar from non-carbohydrate precursors such as pyruvate. In lipid biosynthesis, fats are synthesized using reactions that are the reverse of catabolic reactions. Glycerol is formed from glucose 3-phosphate in glycolysis. Fatty acids are formed by linking acetyl-CoA. Steroid synthesis is more complicated. Amino acids may be synthesized from precursors from glycolysis, the Krebs cycle, the pentose phosphate pathway, and other amino acids. Only some amino acids may be synthesized, and these are referred to as non-essential amino acids. The essential amino acids cannot be synthesized and must be consumed. Amination is the process of adding an amine group to the precursor metabolite. Transamination is the transfer of an amine from one amino acid to another. All transamination reactions involve the use of coenzyme pyridoxal phosphate derived from vitamin B6. Nucleotides are synthesized from precursor metabolites from glycolysis, the Krebs cycle, and the pentose phosphate pathway. Ribose and deoxyribose come from ribose 5-phosphate in the pentose phosphate pathway. The phosphate group is taken from the ATP, and the purines and pyrimidines come from glutamic and aspartic acid derived from the Krebs cycle intermediates. Cells have several ways to regulate metabolic reactions. One way is by synthesizing or degrading channel and transport proteins. This allows the cell to regulate concentrations of the substrates or products. Cells may only synthesize enzymes to catabolize the substrate if the substrate is available, helping to conserve its resources. If two energy resources are available, the cell will use the more energy-efficient source. In some cases, when a cell can synthesize a metabolite, it will cease synthesis when that metabolite is available as a nutrient. Eukaryotic cells can isolate enzymes with membrane-bound organelles. Enzymes can be turned on and off using allosteric sites on the enzymes themselves. Negative feedback loops can shut a pathway down when the product becomes abundant. When pathways use the same substrate, they may be regulated by requiring different coenzymes. When regulating enzyme production by controlling a gene, the cell will control the amount and timing of enzyme production. When controlling metabolic expression, the cell controls the activity of the protein or enzyme after they are already produced.